For those of you who have had my undergraduate evolution course, you'll notice some repetitiveness here. You've seen this before. If you remember it, great. Pat yourself on the back. It'll be a little easier for you. But for those of you who have not, this may be a lot of new material. But again, it's worth reviewing, worth going over. So, what have we learned from all of this DNA sequence that is now available? As we evaluate entire genomes, as we compare them across different species, what do we find? And one of the first and maybe most surprising of the findings was that genomes of organisms vary widely. And there doesn't seem to be any rhyme or reason. There's no clear correlation between features of organisms and their genome size. And this is called the C-value paradox. So basically, your genome size cannot be predicted by an organism's complexity. However we measure complexity, we can't predict genome size, and vice versa. Some very simple genomes, like this amoeba here, have very large genomes. So 670 GB, that's gigabases, 670 billion base pairs of DNA in its haploid genome. The cow is on a par with humans, about a little bit more than 3 billion base pairs of DNA. So this very simple single-celled organism has 200 times as much DNA as the cow. Well, why? What is it? And so you see in this graph here, this is illustrated, that there's some organisms that have large genomes with no rhyme or reason. Salamanders and lungfish do. And then there are others that have a very wide range. And so there's really no prediction. There's no clear trend. Now notice, and I'll just make a note of this, that generally prokaryotes, Parkeria and Archaea, and viruses have smaller genomes than the eukaryotes, but there is some overlap with the protozoa. Okay, so just be aware of that. So the one kind of maybe general thing that we could say that does not follow the C-value paradox is that prokaryotes do almost always have much smaller genomes than eukaryotes. But amongst the eukaryotes, it's very, very clear there is no correlation between complexity of the organism and the size of their genome. Now, we've talked about DNA sequencing. Review that if you need to, but that's where these genomes came from. And we get whole genomes, which is a step forward from targeted sequence, but we can target specific areas if we need to fill in. And then transcriptomes can provide also a step way for identifying genes. And there's a timeline here. You don't need to spend time memorizing it, or even spend time learning it in any really amount of detail. I just kind of wanted to give you an idea. Again, the human genome by draft sequence was in 2001, but other genomes, because of their size, had been done before that. Now, since this, we've now sequenced thousands of genomes for different species, and even within single species like humans, Drosophila, some other medically important species, we have hundreds and hundreds of individual genomes, so we can look at population level trends within those genomes. Now, let's explain the C-value paradox. And it turns out that the C-value paradox is rather easy to explain. There's only one main reason for it, and that is the non-coding DNA. Eukaryotic genomes have huge amounts of non-coding DNA. In the human genome, it's 98.5% of our genome. In others, it's maybe 99.9 something percent, so massive amounts. And so because that non-coding DNA only contributes a very little bit to phenotype, you can get lots and lots of it without having much differences in organisms. And very simple organisms can add lots and lots of non-coding DNA and still remain very, very simple. And so then maybe the better question is, why do we have so much non-coding DNA in these eukaryotes? Well, that's a little tricky to answer, but in short, it comes down to when DNA is added to a genome, there's often very little phenotypic consequence. So it's not good or bad. And so we add a little bit and it's kept. And then we add a little more. And so over time, if the forces for adding DNA, the mutations that add DNA to a genome, if those outweigh the mutations that take it away, then genomes are going to get larger and larger and larger, often with very little impact on the phenotype of the organisms. So we look at those balances, we look at all of them, and we've listed some of these. So these apply to all organisms, not just bacteria. But we can lose DNA sequences that include genes. That's often detrimental. We can make pseudogenes, we can duplicate genes, we can add DNA, so we can increase or decrease genomes. And in many cases, these have very little phenotypic impact, and so some of them are kept, some are lost. And if the things that add DNA outweigh the things that take it away, eventually genomes get bigger. Now, in eukaryotes, there are two more important forces, so put a little note there. If you want to make some predictions or you already know, great. But we'll come back and we'll talk about those two forces here in just a little bit. Now, in addition to the C-value paradox, there's another, maybe a little less strong, but still a trend, a paradox, that is called the G-value paradox. And so people said, okay, if there's really no correlation between complexity and genome size, then let's get rid of the non-coding parts of the genome. Let's only look at those coding parts, that 1.5% in humans, or small percent in another organism. And so they began to do that. And what they realized is that there still wasn't a clear trend, that some much simpler organisms like C. elegans can have larger number of genes and larger number of splicing products, which is kind of what we're looking at, the codes and then the protein coding sequences. So these are the genes themselves and then how many different variants we have. Is that, C. elegans is simpler than Drosophila, but it has more genes, right? Drosophila is simpler than the pufferfish, and so that correlation would make sense. But the pufferfish as a fish is much simpler than mammals, and it's got larger genome by either count than humans. Okay, so there is no clear trend between even the coding sequence. Now, this is a little bit more complex to explain, but there are three main reasons, and you need to know these. The first is that your complexity is based not just on the number of genes, but how those genes are networked together, how complex the interactions are between those genes to give a final product. In addition, you are also not just your coding regions, but how those coding regions are controlled. So the regulatory regions, which are not counted in this G-value measure, are also important for phenotype. And then finally, and you can see this a little bit in this diagram here, that one gene does not equal one product. So notice humans have fewer genes than chicken, for instance, fewer genes than the pufferfish, yet they make more final products. Just as an example, mammal immune genes, the ones that lead to our antibodies, are very, very diverse. And the reason they're diverse is because there's a small number of copies, but they can be spliced together in many, many myriad of different ways to yield many, many different end products. Now, we're going to complete this little unit here by talking finally about viral genomes. Viruses probably have something very similar to the early genome, although maybe bacteria, there's some debate about that. Maybe bacteria are better models for the very first genomes on Earth. But either way, viruses have very, very small genomes. However, they have the most diverse of any genetic material. Viral genomes can be made up of RNA or DNA. They can be single-stranded or double-stranded. We don't see that. So all eukaryotic genomes are DNA. All prokaryotic genomes are DNA. They're all double-stranded, right? But in the viruses, we have this array of different genomes. And we see very little non-coding DNA. In fact, there's so little non-coding DNA that sometimes DNA does double duty. So the same piece of DNA may be part of two different genes, either in overlapping or even in out of reading frame sequence. Okay? And why a small genome? Well, there seems to be strong selection pressure in viruses to having small genomes. And this is because viruses thrive when they can reproduce very, very rapidly. So the smaller you are, even a little bit smaller, you're going to gain an advantage. And when you're trying to quickly copy millions and millions of viral strands of DNA before the cell's immunity or before the cell dies or the immunity kills you off, when you're trying to do that, every little bit helps. So natural selection has streamlined the genomes of viruses to make them little, very, very small, and very dense so that there's almost no non-coding DNA in these viral genomes.